Hi guys, Miss Francis here. To continue our discussion of translation, remember translation is the last part of our protein synthesis movie. So just as transcription can be divided into three stages, translation can also be divided into three stages: initiation, elongation, and termination. So what I want to do is take a look at this image and we'll annotate it and discuss this first step called initiation. So do you see how I've labeled number 1 here? I've got my small ribosomal subunit, and that's going to bind to my messenger RNA. Then I've got this special type of transfer RNA called the initiator transfer RNA. This initiator transfer RNA carries with it methionine, an amino acid. And it's going to attach at the P site, and it's going to attach to the start codon that's found at found on the messenger RNA. Now, this step requires energy, and the energy is used to bring together this large ribosomal subunit with the small ribosomal subunit to form our translation initiation complex. So now let's look at a different image and annotate that to describe the second step of translation, which is called elongation. Elongation is another step that requires energy. So let's first review that we've got an E site, we've got a P site, and we've got an A site within our ribosome. Our first step is codon recognition. So on our messenger RNA, you've got your codons. On your transfer RNA, you'll find your anticodons. So this transfer RNA carries with it an amino acid, and it matches up, the anticodon on the transfer RNA matches up to its complementary codon on the messenger RNA. That's what codon recognition means. Then you've got peptide bonds forming between neighboring amino acids. So do you see how this is a growing chain of amino acids, aka a growing chain of polypeptides? We still have our E site and our P site and our A site, and now we've got our peptide bonds forming between our growing polypeptide chain. The last step of elongation is called translocation, where our transfer RNA is going to shift to the left. So the transfer RNA that was at the A site is going to move to the P site. The transfer RNA that was at the P site is going to now move to the E site. So this transfer RNA that was once at the P site is now at the E site. It doesn't have an amino acid linked to it because we linked it here with a peptide bond to that growing polypeptide chain. And now this amino acid is free to return to the cytoplasm to get another copy of that same amino acid. And now let's look at this image to talk about termination, the last stage of translation. So remember, we still have our E site, we still have our P site, and we still have our A site. Termination occurs when stop codons reach the A site. And we have this release factor that's going to bind to the stop codon and hydrolyze, break the bonds between the polypeptide and its transfer RNA that's at the P site. So this polypeptide is going to be broken from the transfer RNA that's at the P site. And now we have our free polypeptide chain. Here's our free polypeptide chain, and our translation complex disassembles. That is the end of translation. So now what happens? How do we get from the primary structure to whatever happens next? Well, from the primary structure, we get to the secondary structure. From the secondary structure, we get to the tertiary and then the quaternary. But how did we get to the primary structure to begin with? It's the sequence or the order of amino acids that dictates the primary structure, which then dictates the secondary structure, tertiary structure, quaternary structure. But how did we get the sequence in the first place? That came all the way back from the DNA. The DNA provided the instructions. The DNA was transcribed into messenger RNA, which had codons that then were matched complementary with the anticodons on the transfer RNA that carried those amino acids. And then it's the sequence of those amino acids that dictates the structure ultimately. So in order to help the particular structures form, we've got things called chaperones, and they're going to help the protein fold into its particular structure. And then the final protein gets modified by the Golgi and then takes on its function either inside or outside of the cell. And we learned about this when we did cell organelles, so hopefully that's jogging your memory. Now, we can block translation. Why would we want to block translation? As a way of regulating gene expression. Well, why would we want to regulate gene expression? Well, let's say we have a gene that codes for a disease. We don't want to express that gene. We want to stop its expression. But now let's say we have a gene that codes for the repression of that disease. Well, then we would want to use this RNA interference as a way of stopping translation in an attempt to regulate gene expression in eukaryotes, meaning what genes get expressed or turned on and what genes get turned off or not expressed. Another type of RNAi is siRNA, small interfering RNA, that can block the messenger RNA from being translated into a protein. And then you've got microRNA, which just degrades the messenger RNA. This image is an amazing overall picture that captures transcription, RNA processing, and translation, since those are the processes that link DNA sequences to the synthesis of a specific polypeptide chain. I recommend you looking at this image, you've got it in your note packet, and going through and annotating this image. Start with transcription, write out the steps, then go through the steps of RNA processing, and then go through the steps of translation until you ultimately end up with your specific polypeptide chain.